I'm in Munich attending the 17th International Acute Mild Leukemia Conference, and I have delivered a lecture about targeting different layers of the epigenome to achieve leukemia stem cell eradication in AML patients. And the fundamental idea behind this project was that we know that the epigenome is highly complex and composed of several different layers of chemical information, biological information. And up to now, we know that information is important to explain the behavior of leukemias, and that idea has led to the use of early versions of drugs that can target the epigenome to try to treat patients with AML. And indeed, one class of drugs, the DNA-methyltransferase inhibitors, are used routinely in the clinic. And those drugs erase cytosine methylation, which is one of the epigenetic marks. Now, there are also mutations in AML that drive abnormal epigenetic patterning, especially including cytosine methylation, and those include patients that have mutations, for example, in an enzyme called TET2 that is commonly mutated in AML, and its function is lost. And that also has led to the concept of perhaps trying to especially focus on patients like that to deliver this kind of therapy. The fundamental problem is that this approach oversimplifies the epigenome because it's composed of multiple layers. Just hitting one of those layers really is not enough to achieve full reversal of the impact of these kinds of mutations. So to explore this question, what we did is we first developed an organoid system where we can culture primary human AMLs for an extended period of time, because these kinds of drugs that target the epigenome have very slow effects. And so 48-hour or 5-day cultures just aren't enough to be able to determine their functions. You need long-term culture, and working with primary cells is difficult, and so we developed a technology that overcomes that challenge so we can do longer studies, maintaining primary AMLs, proliferating and growing ex vivo for long periods of time. We treated in this manner a cohort of 90 patients that were obtained from a clinical trial in the US from the ECOG cooperative group. And about 52 of these samples were interpretable because they proliferated throughout the entire length of the experiment. And we optimized the dual exposure of these primary AMLs to a DNA-methyltransferase inhibitor. In this case, we used 5-azacitidine, but probably be the same no matter which one you used. And we also used another drug that blocks the activity of LSD1, which is a protein that demethylates histone 3 lysine 4 residues. Now, the relevance of this is that these particular residues are involved in transcriptional activation, and so LSD1 represses genes by doing that. And it particularly functions at gene enhancers, which are elements that are outside of genes but that control nearby gene expression. Whereas the effect of cytosine methylation is most clear at gene promoters. So we treated this cohort of patients and found that there was a subset of patients that responded highly synergistically to the combination. There was broad activity in multiple samples, but the particular patients who had the greatest synergism were those who had mutations in TET2. That finding led us to explore in more detail the impact of the combination therapy on primary TET2 leukemias. And we observed, for example, that unlike giving a DNA-methyltransferase inhibitor alone or an LSD1 inhibitor alone, which although they can debulk leukemias, they do not eradicate the stem cells. We found that the combination therapy does eradicate leukemia stem cells and cause profound reduction in the ability of leukemias to repopulate when transferred from one host to another, which is really what we think is the gold standard for efficacy of an anti-leukemia therapy. Since it's not so hard to debulk those patients with AML, but it is very hard to get rid of the cells that repopulate the disease. And so this approach seemed to do that in primary human TET2 mutant AMLs. Looking under the hood of the cells, the reason why this synergy happens is because it turns out we found that loss of TET2 results in two effects in myeloid cells. One of them is an increase in cytosine methylation at gene promoters, and also at the same time a reduction in 5-hydroxymethylcytosine at gene enhancers, a dual effect. There are two layers being affected independently of each other that are contributing to the effects. The hydroxymethylcytosine is important to activate genes, and cytosine methylation represses genes. And so you have two events that repress genes that are nearby. Now, the loss of hydroxymethylcytosine results in increased binding and activity of LSD1 at enhancers. This is the reason why these patients are highly responsive to LSD1 inhibitors, is because LSD1 is actually driving repression of the enhancers where you've lost hydroxymethylcytosine. And we observed synergistic activation of genes that were affected both by promoter hypermethylation and by enhancer loss of hydroxymethylation with LSD1 gain. Those genes with that pattern responded with a synergistic upregulation of transcription, and mechanistically you could see that the chromatin of those genes was changing, and you're enabling the enhancers to actually loop over and touch the promoters, and that's what's causing the strong gene activation. There are several genes that are in this set of synergistically activated genes that seem to explain the reason why there's a response. One of those genes is called GATA2. There are others as well. But by and large, the summary or the implications are that the combination therapy with these drugs may be especially active for patients who have TET2 mutations. And so we would like to see this combination taken forward to the clinic. There already are clinical trials that are planned or going on with this combination treatment, but we'd like to see this done in the context of enriching for patients who have TET2 mutations to fully see the possible benefit that can be gained by targeting these different layers of the epigenome.